There is a single biggest mistake that every student makes when they first outline the introduction to their first peer-reviewed paper. And in this video I'm going to show you how to avoid this very mistake. My name is Dennis Eckmeyer, I'm a neuroscientist, and after having worked as a researcher for over 10 years, I'm now a freelancer as a science editor and science communicator. With this video series, I'm introducing young students to the art of paper writing for peer-reviewed journals. In the previous videos, we talked about how to write the method section and the results as well as the discussion. So if you haven't seen those videos, just find the playlist on my channel and check them out. The biggest mistake that students make when they are first outlining their papers is to think that the introduction is all about showing off what you know. That's because they're used to that from their thesis writing work. But in fact, the introduction is for something very different. What you really are aiming for is to explain to the reader why you picked your question and why you pick this specific way to answer this question. It is not at all about showing off what you know. The introduction is supposed to lead the reader from the big picture background towards your specific question in a straight, logical sort of story. And what you do there is to narrow down the focus from the big picture over some steps that they need to know down to your conclusion actually. And the best way to achieve this sort of structure is actually to construct the outline backwards. When you start writing your introduction from the big picture, you may end up losing your path and going on tangents quickly. If you start with the end, then it's easier to find a straight path to your big picture idea. So to demonstrate what I mean by constructing the introduction backwards, let's go through the paper that I've so far used for demonstration purposes just to keep it together. Alright, so I conclude the introduction by saying arousal-dependent olfactory memories may alter odor-evoked synaptic activity as peripheral as the first synapse in the main olfactory system. You write that down because that's the conclusion, so that's where you begin outlining at the end. Now imagine an expert in your field trying to understand how you got to that conclusion. What do they need to understand next in order to get why you've done this and how you got this conclusion. And that would basically be the main result of the study. So in this case, we observed a persistent noradrenaline-dependent reduction in the odor response of olfactory sensory neurons. The suppression affected the responses to odors presented between locus coeruleus activation trials equally. Alright, so the logical question now would be, so how did you get to these results? Well, the study design is explained in the previous paragraph. We measured the activation of glomeruli with wide field imaging of intrinsic, optical and fluorescent calcium signals. So why did you choose this specific study design? Well, you're trying to answer a specific question or to test a hypothesis. Does locus coeruleus activation persistently change the activation of olfactory sensory neurons in anesthetized mice? That was my question here. So now, how did you get to that question? Well, it's because there is a gap in the literature that you want to close, because you think it's relevant. And in my case, I would say, according to the literature, noradrenaline, which is produced and released by locus coeruleus, may reach and modulate these neurons, but an effect had not been measured yet. So now the question is, how did you find the gap? So you explain the knowledge that surrounds your topic that shows that there is a gap. In neurons directly connected to the ones we investigated, noradrenaline has known effects. So I write, noradrenaline can acutely inhibit granule cells and thereby transiently disinhibit mitral tufted cells, which may contribute to plastic changes in the main olfactory bulb. So in the literature, it shows that there is a potential for these neurons to be directly affected by noradrenaline. So the gap is that there is no actual data on that. So why is it that the granule cells can inhibit mitral cells? That's the next question that I would have. So I explain. Olfactory sensory neurons and mitral tufted cells form the first synapse of the main olfactory system, etc. And that means that they are connected. That's all. And it's a very early stage in the whole brain processes olfaction process. So now, of course, the reader wants to know why I'm interested in this, and that's because we want to understand memory formation in the olfactory sensory system. So I write, in the main olfactory bulb, noradrenaline is involved in odor learning and discrimination of many stimuli, including social cues. In a very specified journal, this could have been the start of the introduction. However, in this case, I sent it to a general journal, and also the topic involves so many different aspects of neuroscience. So I take yet another step backwards from the issue by saying, sensory responses, cognition and behavior are modulated by noradrenaline. This whole introduction writing is an exercise in clear communication. A lot of scientists do not like the idea of storytelling. When they think of storytelling, they feel like they're being manipulated. But to be fair, all communication is a manipulation. You want the other person to think in a certain way, and that's why you communicate. However, there is a clear difference in selling things and in explaining things. And the introduction is not about selling, it's about explaining. It's not really about hiding weaknesses and overselling the strengths or even making stuff up. The reality, the real science is already a story. And this is how you get people to understand what you're about. And that's what the introduction is. And one part of communicating clearly, obviously, is to keep it straight and to make it one easy to follow, logical story. So in this video I explained to you what I think is a good way to outline such a story, and that is by beginning with the end and then ask systematic questions about whatever you just told yourself. Like, you have a conclusion. How did you reach that conclusion? Well, there's the results. How did you get to the results? Well, there's the study design. How did you get to the study design? Well, you have a specific question that you want to answer, and this study design addresses the question. How did you get to the question? Well, there's a gap in the literature. How did you find the gap in the literature? Well, by reading everything around it. And finally, why did you read everything around it? Well, because it's an interesting topic. So this is how you construct, in my opinion, a very clear and very straightforward storyline for your introduction that people can follow and people are willing to accept as motivation for a study. So the last thing you need to do now is to just flesh out the steps in your outline by adding the necessary information to understand everything, and you're done. If you have any questions or critique or suggestions, please leave them in the comment section below. And if you are interested in a PDF with the information in this video, you can find the link to that in the description to this video, somewhere around here. And if you want to watch the other videos of the series, there is a link to that in the end card. So thank you very much for watching. Have a good day. Bye bye. Okay, so how do I get home?